Okay, so we're going to go through the first video for biology and the first video for flipped. So the good points about flipped are that if you're watching the video and you're taking the note and you need to pause for any moment, you can. If you need to hear a section over again or go over it one more time, you can always rewind and play it back as many times as possible. So at the beginning of most videos, I'm going to try to highlight kind of what your note should have at the end. So for today, you should have the 3 reasons why cells divide, and you should understand the main processes that drive cellular functions. So we all know that we start out as one single fertilized egg cell, but as adults, we're made up of trillions of cells. Now, how we get from 1 to trillions is through a process known as cell division. Now, cells can divide for 3 main reasons. The first reason being reproduction. Now, there are 2 main types of reproduction that occur. We have asexual reproduction, which is basically one parent copying everything about itself, growing, collecting resources to the point where it will just duplicate itself. So the parent is an exact copy, or the offspring is an exact copy of the parent. This is usually referred to as binary fission, and we can see it occurring in things like paramecium, that are single-celled eukaryotic cells, and amoeba, or we can see it occurring in prokaryotic cells like bacteria, E. coli. Okay, now asexual reproduction has its pros and its cons. One pro is you don't have to find a mate. The con, however, is because you're an exact copy of the parent, if conditions change and become less favorable for growth and survival, you may not have any new traits to actually survive in the new environment. Now, our second type of reproduction takes care of that. We have sexual reproduction. So this is where we have 2 parents coming together. We have a fusion of both of the DNA from the parents, so we get an offspring that's slightly different than either parent. Now, the pro about this is because we're getting a combination in DNA, we may have an offspring that has different or new traits that could better suit it to a changing environment, or meaning making it more likely to survive, and we come up with the idea of survival of the fittest. The con, however, for sexual reproduction is that you actually have to find a mate to reproduce with. So cells can divide for reproduction. The second reason why cells divide is for repair. If you think about it, you've all probably got a paper cut or some other boo-boo, and after a couple of days, if you look at where that paper cut was, it's barely noticeable, and that's because your cells regenerate to repair themselves. Now, depending on where the cells are, they could repair themselves at different rates. So it depends on kind of what the cell's function are. So, and this is in multicellular organisms. So for example, if you think of your skin cells, they shed pretty easily because they're exposed to the environment. So they need to repair and replace themselves fairly quickly. However, if we look at something like a blood cell, they really only replace themselves about every 120 days. If we look at another cell in our body, like let's say the cell of our stomach lining, it's exposed to a more harsh environment, the stomach acid contained in our stomach, so they actually replace themselves every 3 days. So again, cells will undergo cell division for repair. Depending on where those cells are, it happens at different rates. Okay, the last reason why cells divide is just for growth. Okay, we are not one giant organism made up of only one cell. Okay, as we grow, we have to add more and more cells. And that's because cells start to become inefficient when they become too big. So what does that mean? Well, for example, if you had waste inside your cell and you started to become too big, that waste could not get out as quickly as it needed. And if too much waste builds up, a cell could die. Similarly, if cells needed a certain amount of nutrients and the nutrients couldn't get into the organelles fast enough, the cell would initially basically starve again and die. So cells are very limited by their size, and they're limited by something known as the surface area to volume ratio. Okay, so cells can only get so big or they become basically not efficient anymore. Okay. Okay, so I'm going to give you an example of surface area to volume ratio. Remember, we want to have the largest surface area to volume ratio. That is the best for cells. So here we have a cube. Let's say each of these sides is 2 centimeters. So if we're calculating surface area, it'd be the area of one of these faces, which is 2 by 2, so 4 centimeters squared. And how many faces do we have in a cube? 6. So 6 faces times 4 centimeters squared is 24 centimeters squared. So that would be the surface area for this cube. If we calculate volume, volume is base times height times width. So 2 times 2 times 2 is 8 cubic centimeters. So if we look at this ratio, 24 to 8, we can reduce it to 3 to 1. Okay, so that's a pretty decent number. Now let's see if we take our cube again, but in this case, we're going to chop it up into little ice cubes. So we've effectively changed our widths of these cubes to 1 by 1 by 1. Okay, but we have 8 of these tiny little cubes. So if we look at our volume, just to show you it's the same volume, we have 8 cubes, so 8 times, and then our base height and width would be 1 by 1 by 1. So 8 times 1 is 8 cubic centimeters. So we have the same amount of volume, but look what happens to the surface area when we cut up the cube here into smaller pieces. So in this case, the surface area of one of our cube's faces would be 1 by 1. So 1 centimeter squared. Now, each of those cubes has 6 faces. So 1 times 6 is 6 centimeters squared. But again, now we have 1, 2, 3, 4, 5, 6, 7, 8. 8 total cubes. So 6 times 8 total cubes is 48 centimeters squared. So the surface area to volume ratio for the small cut up chunk is 48 to 8. And again, if we reduce that, 48 to 8 becomes 6 to 1. Okay, so this has a larger surface area to volume ratio than this one. Okay, 6 to 1 versus 3 to 1. So what does that mean for cells? Well, in the case of ice cubes, we know that as we heat these ice cubes, the one with the larger surface area is going to melt more slowly. That's because there's less heat being able to penetrate the ice cube. So if we actually see them melting, right, the big one is basically going to shrink at a slower rate than the small ones. And we've all had a big chunk of ice and we've all had crushed ice, and the crushed ice will melt a lot faster. So how does that relate to cells though? For example, if we have waste or nutrients that need to get into the cell, if the cell is larger and has a larger surface area to volume, smaller surface area to volume ratio, there's less surface. So it has to travel, those nutrients has to travel further into the cell to get where it needs to get. And it might not reach its destination in time. Whereas if we had a larger surface to area ratio, larger surface area to volume ratio, it can quickly go from outside into the cell. And the same thing is for waste. Again, if waste was building up and it needed to get out, it would take longer for that waste get out of these cells, to get outside of the cell. And again, it might build up too fast and the cell would become inefficient and die, whereas in a smaller cell, it can quickly get out. So the moral of the story is a larger surface area volume makes for happy cells because they're efficient. So the last thing I want to talk about is basically what drives all those cellular functions. What brings in nutrients, what takes out waste, how this occurs in cells. And there's two driving kind of processes that are involved. The first one is known as diffusion. So diffusion can occur in any chemical, and it occurs when a chemical moves from an area of high concentration to areas of low concentration. So what that looks like is, for example, if we had a beaker full of water and we drop some food coloring or dye into it, over time what we should see is those food particles starting to spread out until they're equally spaced throughout the water molecules, and this is known as equilibrium. So this is an example of diffusion. Now note, diffusion can occur with any chemical. Okay, it doesn't necessarily have to be in water. It could be in air, and most of you have probably experienced this. If somebody makes microwave popcorn, you can smell it. For example, if it happens in the cafeteria, you could probably smell it down here in the science pod after about 15 or 20 minutes. So again, it doesn't necessarily have to involve water. It just has to involve a chemical going from a high concentration to a low concentration. The second main function that drives these cellular processes actually does involve water, and it's called osmosis. Now, osmosis is specifically the movement of water molecules across a membrane, and the membrane in question is semi-permeable. Meaning it will permeable. It will only allow certain things through. And it's the movement of water molecules across that membrane to an area of high solute concentration. So water would be the solvent, what you dissolve the solute in. So for example, solute might be concentration of sugar or salt in something. Now the reason why the water molecules move is to try to balance out that concentration of solute. So here's an example. We have water molecules. These are our water molecules. And we have, for example, maybe sugar molecules here. So the sugar molecules can't get across our semi-permeable membrane, but the water molecules can. And what happens over time is these water molecules are trying to make this concentration of sugar the same as on this side. So they will move across the semi-permeable membrane to try to decrease this concentration of solute. And over time, it's going to look like this. So these are the two processes that can drive cell functions. One being diffusion, where it's any chemical moving from high concentration to low. And the second main one is osmosis, where it's only water molecules moving from an area of high solute, moving to an area of high solute to try to reduce the concentration of those solute particles.